is Katarzyna Matczyszyn, and I'm from Wroclaw University of Science and Technology in Poland. And I will introduce you today to the beautiful lyotropic liquid crystals, which are the anisotropic solutions, which you will see are really omnipresent around us. Here on the bottom of the slide, you can see the typical pictures taken by us in our laboratory back in Wroclaw, made of molecules which are able to actually organize into lyotropic liquid crystals. So this is one of the features which made me to work with liquid crystals, because with the polarized light microscopy you can see really beautiful images. So I will, as I said, I will introduce you to lyotropic liquid crystals, then I'll tell you a few words about how they are made, and I would like to underline the biological significance of the lyotropic liquid crystals, as well as their potential or present applications in the real world. The first and the most commonly known, actually, lyotropic liquid crystal is the soap bubble. So the soap is one of the most common molecule which can create those Professor Simone just a minute ago told you about the thermotropic liquid crystals, which are now very popular, because most of the screens we're using nowadays are made of this kind of the molecules. So the thermotropic liquid crystals are the molecules which tend to self-organize into various structures, and depending on the shape of the molecules, we can have various organizations and hence various material properties. So, for example, the elongated molecules will have, with the elongated molecules, we have nematic phases, or the cholesteric phase, as Professor Simone told us. We can have also smectic phases. With more planar molecules, we'll have the discotic phase, which then will organize into nematic, discotic, columnar phases, and so on. If we have the banana-shaped molecules, then they will also create other textures. However, today I would like to focus on the lyotropic liquid crystals, which is a distinctive group of the liquid crystals. So what's the difference? The difference between the thermotropic and lyotropic liquid crystals is how they are made of. So the lyotropic liquid crystals are the two component systems which should have the amphiphyll molecule dissolved in a solvent, and hence the most important feature of the lyotropic liquid crystal is that they are concentration and solvent dependent. The amphiphilic molecule is a molecule which has two parts, two moieties. We call them head, and which is usually hydrophilic, and then we have a tail. So the example of such molecule is, for example, sodium dodecyl sulfate, so traditionally known as soap, which forms the micelles, but also these kind of molecules are various phospholipids which are actually present, for example, in the cell membrane and other structures, as I'll show you. So here we have the example of the lecithin, which tends to organize into the pipe. And again, a beautiful photo taken with the polarized light microscope, showing how this organization can be observed with the once we have such molecule possessing the two parts, the amphiphilic molecule, then those molecules actually tend to self-organize into various structures, such as, for example, micelles. So amphiphilic materials are either insoluble or the molecules dissolve only from the micellar solution. If we take the cross-section of such micelle, you can see the organization of the heads around it and the tails inside the micelles. So the nonpolar chains aggregate together and are effectively removed from the water solvent by the surrounding polar. And micelles occur when the solution is relatively diluted and behaves in a stable in water, and provided the concentration of the surfactant is above the critical concentration. Another possibility of arranging of such molecules is the reverse micelles. So we can have also the structures where the heads stick together and then the chains are out of this composition. So usually such structures can be seen in oil-water mixtures where the amount of water is small and it fills only the void surrounded by the polar head groups. Still, we have here some free space, let's say, however much smaller than for the typical micellar. This phase in which the amphiphilic molecule separates the water from the oil is stable, and in the case of micelles, the water surrounds the micelles, whereas in the case of the reverse micelles, water resides inside the micelles, as these are hydrophilic parts. Just as there are many different types of the thermotropic crystals, as you can see on one of the first slides, there are also several different types of the lyotropic crystal phases. In general, the lyotropic crystal phases have been extensively investigated over the whole concentration range. The main difference is that we need to have a lyotropic liquid crystal, we need not only the molecule of the particular shape, but also we need to adjust the concentration of the molecule in the solution, the solvent, and it also is temperature dependent. Whereas in thermotropic liquid crystal, the organization of the molecules, hence the texture of the liquid crystal, depends only on the temperature, so the movement of the molecules inside the phase. So the three different classes, the main different classes of lyotropic crystal structures are widely recognized. These are the three major types of lyotropic crystals. So we have the hexagonal phase, the cubic phase, and the lamellar phase. However, there are also some intermediate phases depending on actual organization of the micelles into the, or like here, the molecules into various 3D structures. These structures have been classified by X-ray diffraction techniques, and then we can associate this organization with the textures, which can be seen with polarized microscopy. A lamellar lyotropic crystal phase is one of the most important, as it is present, for example, in the cell membrane. This is how the cell membrane is built. So we have those layers of molecules which are like interconnected in the bilayer unit by non-polar chains, where the polar head groups are outside of such a bilayer, and then we can have several layers of such a bilayer unit, and altogether they form the lyotropic liquid crystal phase. So The laminar crystal phase consists of the late arrangement of amphiphilic molecules. The nature of the molecules mean the self-assembly in bilayer has the intertwining of nonpolar chains from oppositely directed molecules. The bilayer thickness is usually up to 30% less than length of all trans molecules, and the nonpolar chains and the water layer thickness is between 1 and 10 nanometers if the water content is up to 50% by weight. But as I told you already, although we call them, for example, lamellar phases, we still can distinguish various organization of the molecules in these layers in between them. So some of the nomenclature is taken directly from the thermotropic crystal classification, and it actually can be also seen out of the polarized light microscopy, which is similar then to those smectic or nematic phases, whereas here the isotropic organization, so there is basically no organization between the distinctive layers, or we have really a true 3D organization of layers. So usually laminar pleiotropic crystal phase exists down to the 50% of the surfactant, and below 50% the laminar phase gives way to the hexagonal pleiotropic crystal phase, or an isotropic micellar solutions. And here are some pictures taken in our laboratory of those molecules. In some cases, the pleiotropic lamellar phase is exhibiting extremely dilute solutions. However, and in general, we are less viscous than, for example, hexagonal pleiotropic liquid crystal, despite the fact that they contain less water. How do they look like? So the hexagonal pleiotropic liquid crystal phase has this arrangement in pillars, I would say. So we have not only the organization of the molecules into such a discotic-like structures, but then they tend to organize into the hexagonal arrangement. So these give a similar birefringent texture when seen with the optical polarized microscopy as the thermotropic hexagonal liquid crystal. And they consist of the micellar cylinders of identified indefinite length packed in the hexagonal arrangement. Usually the diameter of the micellar cylinder is typically 10 to 30% less than twice of the length of the all trans not polar chains. As we can have those micelles also in the form of the reverse micelles, such structures also may self-arrange into the hexagonal phase, and then we have the water inside the micelles and the no-polar chain sticking out of the micelle. So these phases exist, for example, in the oil solution. The spacing between cylinders varies from 1 to 5 nanometers, depending on the amount of the water and the surfactant in the solution. And typically, they contain 30 to 60% of water by weight. And despite this high water content, the phase is very viscous. So the viscosity of the hexagonal phase means that it's best avoided applications. And the reverse hexagonal phase is basically the same as the hexagonal phase, but just the difference is that we have different arrangement of the molecules, and the textures which can be observed with the polarized light microscope are as here. In our laboratory, we study quite a lot the liquid crystalline organization of the DNA, mostly in columnar hexagonal phase. However, DNA has actually ability also to self-organize into the cholesteric phase, which was also mentioned by Professor Simoni. However, here we have the lyotropic crystal. So the liquid crystal formation of the DNA chains depends strongly, as in all lyotropic liquid crystals, on the temperature and, more importantly, on the concentration of the DNA. So in the decondensed chromosomes, it will self-organize into the pre-cholesteric phase, whereas in mitochondrial DNA, we can already observe this spherulites and the cholesteric phase appearance. And in the chromosomes itself, we would have the DNA organized in the columnar hexagonal phase. The existence of the specific liquid crystal phases depends here on the temperature and enzyme concentration, but also on the ionic strength of the solution and on the present counterions, as it actually affects the stiffness of the DNA molecule, and it then applies also on the counter and persistence length of these chains. And we also have seen that the sequence and the topology of the DNA, for example, in plasmid DNA, has the impact on which phase will be more. So here I wanted to share some also the pictures, how beautiful these phases can be. So here in the concentration as low as 50 milligrams of DNA per milliliter of water, already the spherulites are present with the increasing concentration. More and more dense phases are observed, and here we have the columnar hexagonal phase. So one of the when I see these photos, one of my thought was how really beautiful inside we are, as all our cells actually contain such arrangement, and the inside beauty actually has a real meaning, not only on our character, but on the can be seen with real pictures, right? So here with even more increased concentration, we can clearly see the columnar hexagonal phase. What's the importance of this lyotropic liquid crystals made by the DNA? So far we know that it's absolutely crucial for the packing of the DNA in vivo. So without this ability of forming lyotropic liquid crystal, it wouldn't be actually possible to fill in all this material in the nucleus, the chromosomes, into very small structure, with actually remembering the position of each base pair in the nucleus, because they are also responsible for the prebiotic chemical generation of the H-bonded DNA assemblies. And we've actually worked with Professor Malcolm Buckle from École Normale Supérieure in Cachan on comparing the organized DNA in the liquid crystal phase and this which was not organized at the same concentration, playing with various counter ions concentration, and we were able to show that if the DNA hasn't been organized into the lyotropic crystal, the process of expressing of DNA changes enormously, with a lot of faults on its expression. However, here we are at the materials science meeting, so the potential application of such lyotropic crystals is also important and can be further explored. as it's a material for low-loss photonic waveguides and devices. We can get optically active materials while we will dope it with chromophores or luminophores. So we are working with azobenzene, for example, and we try to change, so influence the stability of these phases, as well as we are able to get the self-assembled system while with the, for example, aligned nanoparticles. So we play a lot with plasmonic anisotropic structures, as then we can change, depending on the light polarization, for example, we may change the property of just an example of the DNA doped with gold, silver gold nanoclusters, so super small nanoparticles, which has a diameter up to 2 nanometers. And I was absolutely convinced that these nanostructures will go into this phase, liquid crystal phase, and it didn't happen. We observed this organization of these tiny nanoclusters, mostly on the surface of our samples, which was observed with the photon polarized light microscopy. When we doped the DNA hexagonal phase with gold mini nanorods, and we used 750 nanometers light for observations. You could see the luminescence coming out of these nanorods, mostly inside, from inside of the liquid crystal cell. So it means that they actually entered into the liquid crystal cells. So then we have the proof that organization of the nanoparticles inside the liquid crystal cell depends on many factors, but also on the size, and not also very important on the ligands. But this is another story. While working with gold nanoparticles embedded into the DNA liquid crystal, we've seen an interesting feature that while working, like trying to observe a single nanoparticle, we've seen it disappearing after only 2 minutes and 50 microwatts laser power. So this is the polar graph where we've seen this disappearance of it, and we've tried it several places, and each time it's the same story. So we concluded once we had a very thin layer of the DNA liquid crystal, that actually we are able to move around this nanoparticle, which means we actually have here a local phase transition of the liquid crystal, which changes the ability, the fluidity of the system, and the ability of the nanoparticle to move around. So the combination of light and the plasmonic properties of the nanoparticle gave us the way of moving the nanoparticle inside the liquid crystal phase of the DNA. Okay, next arrangement, which I would like to also mention to you today, within the lyotropic liquid crystals, the cubic lyotropic phase. They are not as common as the lamellar or hexagonal phase. However, and they are not as well characterized. However, still, I wanted you to know that they exist, and there are 2 types of cubic lyotropic crystal, and they can be arranged in the manner, again, water continuous or the reversed manner, depends on the if we have micelle-like solution or the reversed micelle solution. So the most well-known cubic phase consists of the cubic arrangement of molecular aggregates, and the molecular aggregates are similar to micelles or reverse micelles. Some reports suggest the molecular aggregates are spherical, but others claim they are cylindrical or ellipsoidal. Cubic lyotropic liquid crystals are extremely viscous and even more viscous than the hexagonal phase, and sometimes they're called viscous isotropic phases. In nature, makes them difficult to be detected by polarized light microscopy, and that's why sometimes they just remain undetected, as polarized light microscopy is one of the main techniques of observing however, here you can see how of the photographs of the cubic texture arrangement. As I told you already, the lyotropic liquid crystals are known of having this appearance depending on the temperature and the concentration of a molecule in a solvent. So one of the examples how it looks like, how such a phase diagram looks like, is here. So first we have just the micelles at the low concentration. I will have just the micelles in the isotropic solutions. However, with the rise of the concentration and here also with the temperature, the micelles tend to organize in the hexagonal cubic phase here. Then we can have the hexagonal arrangement and then more complex structures such as, for example, here, lamellar structure. So with the same type of molecules playing with its concentration and temperature, we can get different arrangement freely in the three pictures. So phase diagram are constructed with the amphiphilic concentration along the horizontal axis and temperature along the vertical. And such phase diagrams are often used to show the liquid crystal-based behavior of a mixture of sometimes two thermotropic liquids. And this is a real picture of how such phase diagram look like in real life, right? So various organization appears depending on the concentration, and with the polarized light microscopy, it can also be observed. So here we have, when it's black with the cross polarizer, we have just isotropic solution. It's the beginning of lamellar isotropic phase, a typical lamellar nematic phase, a gay lamellar isotropic phase, and the columnar hexagonal phase. Lyotropic liquid crystal polymers are the materials which I wanted to mention, because myself I was really surprised some time ago how they look like and where they can be seen. So the polymer is to be, if a polymer is to be a lyotropic liquid crystal, it must firstly be fairly rigid, and secondly it must dissolve in a solvent. So usually, being a chemist, these requirements are often mutually exclusive, as if something is rigid and probably it's not well soluble. So accordingly, a drastic solvents such as sulfuric acid, for example, are required. However, for example, Kevlar is one of such materials, which is a synthetic polyamide of a simple structure. But when dissolved in high concentration in sulfuric acid, it has the liquid crystalline phases. So this ability of forming this liquid crystal phase by this polymer makes Kevlar like super strong material. For example, compared with nylon, it is 30 times stronger, despite being only slightly more dense. In fact, steel is 5 times more dense than Kevlar, which kilo for kilo makes Kevlar stronger than steel. And this ability comes actually from its organization into the liquid crystal phase, so that it possesses the 3D structure, so it is able to absorb more shock than the crystal-like structure. So it's more elastic, although at the room temperature and without the solvent, we've seen it as a solid material, right? The Kevlar is not flowing over the body of the police, for example. Other materials, for example, so the bulletproof vest, but also diving gloves, walking boots, fireproof clothing, and other materials are also known to have the ability of self-organizing pleiotropic liquid crystals. However, so this is kind of a distraction. However, as I mentioned to you, from my point of view, the most important feature of the pleiotropic liquid crystal is the behavior in biology, so of the presence in biological structures, such as, for example, biological membranes. So the plasma membrane of cells are constructed of the phospholipids, so we have the polar region and the non-polar region, and they closely resemble of the soap and detergents molecules, and hence they have the ability to self-organize. You can see what we know from the biology lessons is the bilayer of the phospholipids, where we have also integrated some proteins, for example, which are incorporated through the membrane, or which are just peripheral proteins. And this ability of self-organize into the bilayer gives this possibility of the cell to be at the one time a very distinctive moiety, very distinctive structure in our body, but at the same time being very well permeable for the compounds we want to, that our body wants to be permeable to, and strong enough to keep other cell elements inside the substructure. So the soap bubble is a parable working analogy to the cell membrane. So both the plasma membrane that encases the cell and the intracellular membranes that bound our cellular organelles, such as nuclei, and the mitochondria exhibit some liquid-based space enclosing behavior. So I suppose that after this lecture, you won't look when, for example, washing your hands at the soap in the same way, because it's a really very well organized structure, which resembles the internal. So apart from being already present in our body, naturally present, so the really incredible achievement of the biology, we may also use this self-organization of the pleiotropic crystals into liposomes, for example. So some drugs which are insoluble in blood and will be not very effective. By the addition of the small quantity of amphiphil can help solubilize the drug molecules, and they can be either put inside this inside the structure, or they can be encased inside the hydrophilic core. So the liposomes can serve us as a way of delivery of the drug. They can be either attached on the structure of the liposomes, they can be inside, like the hydrophobic drugs, or they can be inside, including small fragments of the DNA, RNA, and other molecules. And depending on the targeting ligand where we want to go, we may specifically change. As I already told you, one of the reasons why I'm working at all with lithographic crystals is the beauty. So here is the example of some liposomes. So these are giant liposomes taken by the people in Denmark, still only with, this is the confocal image, but still you can see how beautiful they are. Lithographic crystals are, or I would say, disappearance of such arrangement, unfortunately, might cause some of the diseases. So some disruption of the biological electroplating crystals can have disastrous consequences. Such a disruption has been tracked back to many diseases. So among other subjects, we are working also on the amyloid distribution, which causes for example, Alzheimer's disease or the Parkinson's disease. So it has been noted that the symptoms appear when the lithographic crystal phase of the amyloid is disturbed. And this is very interesting because the scientists, don't know yet if the fibrillous aggregations of the plaque aggregation of the amyloids causes these diseases, or actually we are, because it's usually full of also toxins or heavy metals, or actually this is the way of the brain or the kidney or other organs to protect it from distribution of these toxins into the body. However, as you know, the consequences are pretty devastating. So this is one of the photos how it is observed. So the plaque formation of the proteins, which then can be detected. How we studied. This is a very simple example of the bovine insulin, which we want to mimic the in vivo conditions. So as you can see, just this is a native picture of the native structure. However, if we incubate this protein with the low pH, which by the way is present while signaling the nerves signals in our brain, we can see the maltase process, right? Which are one of the features of the liquid crystal, for example. And the CM picture is this organization into the spherulites, right? So we have the spherulites, which when I first saw this picture taken by one of my students, I thought about this zeolite the Katarzyna, and I have to admit that really it resembles one of this fruit on this. Really, it does look like it once imaged. So nowadays we try to influence the stabilization. So we try to destabilize this structure either by light or by adding gold nanoparticles. So we can heat up inside the structure. So to get rid of this, because this is a crystalline structure, right? This is a crystal-like structure, and we want to go back to liquid crystal crystallinity of this structure, or we might also add some photochromic molecules eventually to also disturb this. A lot of our work is also concentrated onto the myelin properties. So multiple sclerosis is another disease we are interested in. So it's characterized by local disintegration of the myelin sheet around the nervous axons, which results in the electrical signals being impeded as they travel along the axon. The myelin sheet is a phospholipid bilayer, which acts normally as an insulator. So in the healthy nerve, the myelin sheet is intact, and the signal may travel without any disruption. Whereas when the demyelination process is in progress, we have the signal processing. So the myelin is made of stacked lipid bilayers. We usually have several of these bilayers around the axon. Here while studying with the polarized light microscopy, the microscopy, we doped it with the red Nile molecules to have a better image. Right. So here we have this destruction of the nerve, so you can see the poles do not reach the end of the axon, whereas in the healthy nerve it may travel all along it. And our Nile blue, Nile red molecules are just in between image the structures. Polarized light microscopy, when we grow the myelin in the in vitro condition, so we have the phospholipid mixture with the condition mimicking the real concentrations and the real condition which appear in the brain, and we can see the formation of this in layers. They sometimes are not only the elongated structure, but they also tend to curve, or sometimes they have other shapes, and depending on the condition we apply to, we can have the looped structures of the solid structure. They can be hollow or oval-like. We may have the spoon-like structures or like structures. So simply by extracting this phospholipid, we are not only studying its behavior in a biological context, but also from the material science context. What kind of other interesting organization can we get while playing with external conditions on such? We also dope these structures with nanoparticles. These are microparticles, actually, with silicon dioxide. So when I saw first this picture made by Dominika, first about the lizard skin, but then it looks really like octopus structure, right? So maybe we may also play with this, by the way, amazing animal. We dope the sheets with, for example, super bright carbon dots, so we may get the material which consists of myelin, so phospholipids and organized carbon dots here in this effect. So carbon dots may serve both as a marker because they are completely non-toxic. We did such studies. Or we may again have a new material available for material science studies. So the disappearance of the myelin sheet is not only because does not only occur in multiple sclerosis, but also other diseases. And what we would like to achieve with doping the myelin with nanoparticles is actually trying to have the to induce the remyelination of the axon, so the rebuilding of the phospholipid layer by keeping it liquid crystallinity. Right. Okay, so I told you already about the ability of the DNA to form crystal phases, but how can we see it, for example, in the chromosomes, right? So this is the picture of the chromosome IFM, but CM picture. What we can observe here is actually just the surface, and what I wanted to see was how we could play with this liquid crystallinity arrangement inside, for example, the chromosome. Here we have the two-photon microscopy, which comes as a natural tool for these studies. So in one-photon microscopy, we have the light which will go through all the sample, whereas when we apply femtosecond lasers, and we make use of the multi-photon processes, so 2-photon fluorescence, for example. We have just a very confined light spot, which by traveling through the sample can give us the information about a very local arrangement, including inside, for example, the chromosome or inside the myelin sheet. It has definitely much better resolution. It is much less damaging, and we avoid the problem with photo bleaching. Just one very slight inconvenience is that we need to use the femtosecond lasers, which are usually very expensive. However, if we take if we build up in our system also the polarizers, we may also have the answer about the organization of our molecules in the systems. We may analyze, so first we polarize the light which induces the fluorescence of our sample, and then we also analyze the response in a manner of light polarization, and then depending on the response we may have the information about the orientation of emitting dipoles. So here is the example of the myelin sheet studied in this manner, doped with the carbon nanodots. So we could see the distribution of the carbon nanodots in the 3D manner. I would say inside such. We also dope the neurons with gold nanoparticles and studied it also by the 2-photon microscopy, which can be seen. It's actually very. Okay, so just briefly to say that it's not only what we are working on, so the DNA or amyloids or the myelin sheets, but also for example, sickle cell anemia is one. It happens because these cells, erythrocytes, are losing their liquid crystallinity. So, one of the reasons when the cholesterol starts to build up in the veins, also it's not a liquid crystal anymore. It may cause the clogging of the. And last but not the least, the Nano-fabrication crystal have also a lot of industrial applications, such as, for example, of course, soaps and detergents. And interestingly, washing is a relatively expensive business. So if we are able to find something which form a Nano-fabrication crystal phase at the low temperature and with the low concentration, that somebody could make a lot of. If we're talking about the money, we should also talk about the oil industry. So Nano-fabrication crystal also are used, for example, to take off the remaining oil in the rock, so they can be extracted with the use of Nano-fabrication crystal from the pores. Probably also you did not connect the Nano-fabrication crystals for the so all the mayonnaise or salad dressing, marshmallow, so on and so forth. Actually, emulsifiers in general are lyotropic liquid crystals. Some grains are used for making bread have natural amphifields, which serves as for food emulsifiers. Other are. This is the example which I think is absolutely a masterpiece done by L'Oreal a few years back. So I probably most of you came across the micellar solutions for example, face washing and stuff like that. So what did L'Oreal was absolutely brilliant. So they just dissolved soap, and they sell 2% soap solutions with some additives, but like polyglycols, for the price which is 10 times more expensive than the price of the soap. Absolutely brilliant. You have a product which, first of all, uses less of the material. Second of all, is by price to price is about 10 times more expensive. But if you take into account the 2%, up to 2% concentration of the soap, has like a 1,000% profit on it.
There is a bubble also. The bonds and the bubble which compensate the cost and explain why it's so expensive.
Yeah, so myself, I cannot use it. I prefer the soap with some eventual additive. Final comment is lithotropic crystals were actually the first liquid crystals to be discovered. Much research has been carried out on lithotropic liquid crystal because of the commercial potential in household products and food. However, the importance of lithotropic liquid crystals has been completely overshadowed by the thermotropic liquid crystals because those caught the imagination of researchers at the right time and are tremendously successful in display devices, from simple watch and calculator displays to very large, intricate color television displays. Still, it caught my attention because of its beauty and because of its biology. So, lithotropic liquid crystals are connected with things that are taken for granted and not seen as technologically important. And in my opinion, it's a pity because research into lithotropic liquid crystals still has much potential. So far, very little work has gone into design and synthesis of novel lithotropic materials, and potential uses have not been extensively investigated. There will always be a need for better, more effective detergents that, for example, act at a much lower temperature, less damage to delicate items. Most importantly, life itself is based on lithotropic liquid crystal systems, and it seems certain that future biomedical research will include studies into lithotropic liquid crystalline structures of life systems. So, in my opinion, overall, lithotropic liquid crystals look to have a very significant future. After all, we know how important life is. And thank you for your attention. Cheers. This is the soap, and some soap with the polarized optical. The liquid crystalline DNA was the cover of Nature magazine because some pictures were sold on the auction for the children with autism. This is how the phase transition, various phases of DNA.